Well, that was a good introduction. Thank you. And apologies this year again. I will not give my lecture in Portuguese. But it's impressive to hear how certain people are coming on with their accents. And I can actually understand Mario Stevenson better in Portuguese. So congratulations, Mario. And apologies for these the technical issues. I have a couple of videos that I wanted to show. And I've just got one of these new MacBook Pro from Apple, which comes with a whole pile of things that they sell you that you didn't need before. Nothing works with anything else. So you had a couple of minutes to digest Mario's incredible lecture, because he really set my lecture up in a way that I both anticipated and was grateful to hear. So what I'm going to do is talk about a new venture, a new grant that we have recently been awarded. And this is called Believe. Believe. And this stands for something. Bench-to-bed enhanced lymphocyte infusions to engineer viral eradication. And Brad Jones, who's an assistant professor at George Washington University, came up with this. Catherine Bollard, Brad, and myself were sitting around early in the morning drinking coffee, trying to come up with a name. And we think that this really tries to encompass what we'd like to do to get rid, if we can, of the reservoir that Mario so beautifully described is going to be incredibly difficult to do. We were funded, or are funded, in this 5-year grant by the National Institutes of Health. And this is one of the Martin Delaney Collaboratories for HIV cure research. Martin Delaney was an activist who I met several times when I was in San Francisco, and he was one of those inspiring people who constantly told us scientists we had to work harder and quicker. And he would not take no for an answer, and he constantly asked us to push things towards people. And that had a very big impact on me. And so in leading this Martin Delaney Collaboratory, we have made sure that we plan within this grant to try and test our products in people. It's a large grant, and therefore there are a lot of people involved. It is a rather unique mechanism by the NIH called a UM1 grant, and that means we have very close connection with our project officers, Lillian Kuo in particular. And there are, as I'll show, a large number of different sites at which we are working with scientists on this project. And I'll talk about the goal in a minute. So we think ourselves as an Americas-focused group. We're based out of the George Washington University in Washington, D.C., and we have several sites in both the US and Canada, but also in Mexico City and down here at the University of Sao Paulo. We have leadership structures. So I'm up there trying to direct all these people. We have an executive committee. We have a strong group of community engagement. I run this with Mark O'Callaghan, Robert Furler. And then I'm going to talk about the work in each of these initial research foci. And then we are supported by different support sections. One of the genius things about this grant is that they have made us, and we wish to, collaborate with companies who have products. And so we are working with Altor Biosciences and Talk, and I'll tell you a little bit about them. So this is based out of the George Washington University in the capital city of the United States. And we are located here. And we are 2 miles away from this white building, the White House. And we hope to be able to influence in a positive way Okay, yeah, go on. So if we can't do that, we're very fortunate in being close to the mall. It's a beautiful part of the city. We can go and reflect what would Lincoln have done in face of challenges. We're not sure which is the bigger challenge, trying to cure HIV or... So Washington, D.C. Has a large number of HIV-infected people living in it. In fact, it's one of the worst places to live in the United States for HIV infection. If you look at the numbers, there are around 13,500 people who are living in this reasonably small city with HIV infection. There have been good plans and progress to try and reduce the number of newly diagnosed, lots of prevention activities, and those numbers falling are great. But if you look at the demographics of who's infected in the city, it is still, to me, continuously shocking that we are living with more than 4.5% of black males in the city with HIV infection. If you look at Hispanic males, black females, white males, also around 2%. And these are sub-Saharan African levels of HIV infection, just a few steps away from the White House door. Someone who is a young black male who lives in Washington, D.C., has a 1 in 2 lifetime chance of becoming infected. And of course, that makes the epidemic close to home and something that, with our Delaney, we're hoping to contribute towards. Altor is our first company that I'm going to tell you about. It's led by a very experienced CEO, Hing Wong, and they've been focusing on discovering novel cytokine-based immunotherapeutics. It's based in Florida, and they have a large portfolio of patents and 8 active clinical studies, including 1 in HIV, 7 in a number of different malignancies, as well as in combination of their products with some of these amazing oncology antibodies that are transforming cancer therapy. And we are using, and in fact, Tim Shacker, who's talking at this course next week, also has been using this molecule ALT-803. And so far, over 100 patients have received that. And this is, if you like, a souped-up IL-15 molecule. It's a superagonist fusion complex where there's improved binding activity engineered here, and compared to IL-15 without this complex, it's 30 times more active. It induces T cells to produce cytokines such as interferon gamma. It's got a longer half-life, and I'm going to show you some brand new unpublished data which suggests that this also can help drive these cells into lymphoid tissues. So Brad Jones is part of the Collaboratory, and James Whitney, also part of our Collaboratory, have shown in vitro that ALT-803, this IL-15 superagonist, can also act as a latency reversal molecule, both in a primary cell model and also looking at patient samples ex vivo or in SIV-infected primates. So Alt-803 is not only an IL-15 supercharged molecule, it can also act in the context of a lentiviral infection with reversing latency. Talk is a biotechnology spin-off from MIT in Boston, and they have this backpack cell therapy. It's like, you know, we all go around with our backpacks on. They have worked out how to molecularly conjugate backpacks onto cells, and those cells will run around the body with a backpack on them, and that backpack can be loaded up with a number of different products. And they have not yet got a product in clinical study, but they are working towards that. These backpacks are loaded up. For example, we could put this Alt-803 molecule into the molecular backpack and then inject that, and it will go where those cells go. Brad Jones took this picture. I hope we can see it. The backpacks are in blue. You have to be quick to catch them, but they are tagged on to cells, and they stay there until they release their cargo. We believe that community engagement is really important, and we have a strong community engagement group. We have an advisory board. We have members of the community sitting on this advisory board, and they provide feedback on protocols and ethical concerns, and also develop outreach and educational materials. And in fact, this has already begun here in Brazil with Esper and Camilla. I was a little worried here because I realized that we've nibbled off some of the order and progress in the Brazilian flag, so apologies for that. So that's the concept. That's what we are setting up. What do we want to do? Well, to try and keep it reasonably straightforward, we want to take HIV-infected people's lymphocytes ex vivo and make them work better, and then infuse them back in combination with latency reversing agents. And we really want to make sure that these souped up effector cells get to the place where the virus is hiding. Now, we've heard from Mario that that's very complicated, particularly when it comes to the macrophage reservoir, but we would like to try this approach. And if you sum this up in 4 words, it's enhanced targeted cell therapy. So that is the scientific goal of this Believe Collaboratory. So I mentioned that we have 4 scientific structures and 3 support sections. So over the next few minutes, I'm just going to tell you some of the newest work that we are doing in each of these scientific foci to give you an overview of where we are and where we're trying to go. So the first initial research foci is co-led by Brad Jones and myself. And this research program aims to harness natural and engineered cytotoxic T cells to attempt to eradicate HIV reservoirs. And we are modifying T cells in a number of different ways. So I'm going to show you some new data generated in collaboration with Ed Berger at the NIH, who is part of our collaboratory, on bispecific CD4 positive T cell CARs, these fancy chimeric antigen receptor effector cells, which are really now being used quite extensively in the cancer field. We're trying the same approach against CD4 positive HIV infected cells. We are also working with HIV-specific cytotoxic T cells, where we know which epitopes they target, and using different ways of expanding them. And then we are testing each of these approaches with or without latency reversing agents, such as Alt-803, and also NEF inhibitors. And we are aiming and have started in preclinical studies and studies in non-human primates to give them either systemically or via those little backpack CTLs that I showed you the slide of. So what we're really trying to do is to sort of apply the shock and kill strategy to individuals who have suppressed viremia on long-term antiretroviral therapy. And so the question is, can these CTL clones, in combination with a molecule like Alt-803, drive reductions in the natural viral reservoir? And so we are planning and are in the process of testing many different combinations of latency-reversing agents with CTL clones. And we start out in vitro, and we use what Brad has coined a hive assay, or an HIV eradication assay. And we've tested a lot of different LRAs. We've tested various immune enhancers. We've looked at alternative immune effectors. And the scientific rationale for this part is really to try and understand whether we can get rid of reservoir-infected cells by CTL or not. So this hive assay, we take leukapheresis samples from HIV-infected participants who are on antiretroviral drugs. We take out the CD4 cells. We have a lot of them, and you need a lot for this assay. And then you add HIV-specific CTL with or without latency-reversing agents. You co-culture with ARVs, and then you look after depletion of CD8 cells with readouts of purity, activation status, looking at the amount of HIV DNA. You heard a lot about that from Mario, cell-associated DNA, which we use with our droplet digital PCR machine, and then a couple of enthusiastic graduate students who like doing quantitative viral outgrowth assays, or QVOAs. And so we get a number of different measurements from this hive assay. And the first good piece of news is that in this assay, we can take from a QVOA, single QVOA wells, and infect those activated CD4 T cells, do this co-culture, and then add in autologous CTL, and find that those CTL do recognize the HIV-infected cells. And when you add in the CTL, you eliminate HIV-infected cells. So this is a virus elimination in vitro assay. We can use these CTL to get rid of that activated virus-infected cells. So what might be the barriers that prevent CTLs, which are already in patients, from actually getting rid of the reservoir in the first place? Well, there are a number of potential barriers that we can think of, and also potential solutions. So one important one is sequence diversity and immune escape. And a potential solution is to boost responses against non-escaped epitopes. Also, we've heard a lot and consider how these T cells, they're doing a really tough job, and they get tired, they get exhausted. So T-cell exhaustion or dysfunction, we could think about cytokine therapy or checkpoint inhibitors. It may be that the virus is hiding in places that the CTLs can't get to. So we're interested in redirecting CTL to try and get to those follicles, and also potentially disrupting the follicles. And then thinking more about what Mario was talking about with the latent population, can we have a shock and kill approach that allows these CTL to come in? So the problem is that so far we've tested all of these, and we still can't get rid of the inducible virus that we outgrow from a viral reservoir. And so what we're seeing are resistance to killing by CTL in the natural reservoir. We can use CTL to reduce virus DNA, no problem. But we can't get rid of virus that is outgrowing from the inducible reservoir. And so, there might be some reasons for this. One that we thought of was that perhaps there was increased expression of exhaustion markers in the presence of some of these activating agents. We know that NEF downregulates MHC class 1, and that could potentially inhibit CTL recognition of virus-infected cells when NEF is intact. And so part of the Collaboratory, and I'll come on to, is using small molecule NEF inhibitors to try and block that. Another approach is we could target non-MHC class 1 restricted antigens. We know that Jonas Sasha in Oregon has really developed an interesting story in relation to non-classical T-cell recognition in the SIV model, and that's something we are actively working on. And then what we're worried about is that HIV, there are a lot of defective proviruses. Not every virus is intact and viable, but there's a lot of defective viruses around, and we think they could potentially act as decoys and taking the CTLs away from where they really need to go. And so one thing is that we could potentially increase the effector-to-target ratio. So working with Ed Berger, we have used a number of his CD4 positive CAR CTL. So these CARs can redirect CTL responses against non-TCR targets. It's now a reasonably standard molecular approach and has been used in the cancer field in the United States now quite extensively. And Ed and his lab developed these CARs that could target CD4 positive cells, and they have now come up with new ones which will have enhanced binding to GP120 but can't be infected by HIV themselves. So they're CD4 positive effector CARs. And once that CAR is activated, then you get triggering and killing. So if you look in a recognition assay, does one of these CD4 CARs degranulate and produce cytokine in response to autologous HIV-infected targets? So we've got controls, CAR alone, mock targets, and then targets that are infected with virus. And yes, that CD4 positive CAR T cell specifically recognizes HIV-infected targets. And then I've already shown you data from an elimination assay in vitro, and so using the CD4 CAR in an elimination assay, we see that if you increase the number of CAR effector cells compared to the number of targets, that you actually can eliminate HIV from a culture by using the CD4 CAR T cells. That's a fantastic result because that suggests that this therapy that is now routine in cancer, by using these modified CD4 positive CARs, if we infuse those into HIV-infected participants, then maybe we would be able to reduce viral load even more. So what we've really been trying to find out is, can CTLs in combination with LRIs drive reductions of the natural viral reservoirs? And we've shown that CTL clones can deplete cell-associated HIV DNA. And the eliminated cells are most likely to contain defector provirus as the level of the infectious virus in the cuvera remains constant. But the important thing here, and this is real science, real data, we're currently stuck in trying to get rid of the infectious reservoir. So how can we potentially overcome this barrier? So Yaoqi Ho in Bob Silicano's lab has really elegantly defined where virus comes from in people who are on suppressive heart regimes. And you have this very large pie of defective provirus. You've got a smaller pi of intact, non-induced provirus. And then the infectious virus is really coming from a small reservoir. And so we think that there's a group of defective proviruses which don't ever express antigen, but also that there are inducible defective proviruses which can be made to produce antigen, and that's presented by MHC complexes on the surface of the cell. And then, for reasons which we don't understand at the moment, we don't get targeting of the intact inducible proviruses, but you can take viruses from these and put it into newly infected cells, and they get killed just as well. So this is sort of a puzzle to us, and we're working very actively to try and overcome that barrier of CTL recognition of the inducible reservoir. So the next compartment, I had talked about how the HIV-NEF protein has a number of different characteristics, one of which is to downregulate MHC class 1. So another potential approach to make the CTLs work better is to design chemically molecules which might be able to inhibit NEF. And Tom Smithgull in Pittsburgh is our believed collaborator who's been working on a number of different collaborators. This is a molecule called B9, and it's predicted to make contacts with certain NEF residues, and we are in the process of testing this in vitro to see whether it will improve the CTL killing. And then we've got one publication. We've just been going for 5 months, and we're building up our studies as we go. So IRF2 wants to do something a little different. The first scientific focus was to look at cytotoxic T cells. The second focus is to combine broadly neutralizing antibodies with the super IL-15 agonists Alt-803 and LRAs to mobilize natural killer cells. Natural killer cells are also potential agents, effectors, which could mediate antibody-dependent cellular cytotoxicity. And so we're also in the process of modifying NK cells to augment their capacity to mediate ADCC. So, for example, we have been generating natural killer cells, which actually we've engineered to produce broadly neutralizing antibodies. So this little NK cell can go around, and if it engages a target, it also secretes a neutralizing antibody at that local site. And some of these we're still working on. So Harris Goldstein and Einstein in New York, who's doing this, takes leukapheresis samples, quantifies the reservoir, screens donors, and then he's got a little mouse model with immunodeficient mice. He puts in PBMCs from these ARV-treated participants, and he monitors viral loads, and then he uses this model to study and evaluate these different conditions. So if you like, it's one model removed from the in vitro. It's heading towards allowing us to be able to decide which we want to take into non-human primates. But Brad and Yaoqing, his postdoc, one of his postdocs in the lab at GW, thought that it was probably really important, if we're going to put in neutralizing antibodies, to try and understand which neutralizing antibodies could actually bind in the system. And so we've got a large number of antibodies, many of which have gone into people, if not all of them have gone into people, and we conjugated them and used them in a binding assay, because in order to have ADCC, you'd need to have that antibody bind. And so this is one donor, OM5001, and testing 3 different antibodies. And in this case, we see PG9 binding to infected cells better than the others. But in another individual, CIRC0133, we found that actually 1074 bound better than the others. And so what we're finding is that different reservoirs and different individuals have different ways that they bind to these antibodies. But when we break it down by where there's broadly neutralizing antibodies target, we see a few patterns. So in the CD4 binding site antibodies, we found that no CD4 binding site antibodies bound to HIV-infected cells. And the same was true for GP41. So no GP41 neutralizing antibodies bound to HIV-infected cells. When we came to the V3, glycan, and V1, V2, we did see some high binding capacity, but also some non-specific binding as well. So we found that these broadly neutralizing antibodies have different binding properties from different donors, and the PGT121, 1074, the best binding neutralizing antibodies that will potentially be useful in ADCC. But not much recognition with the other antibodies. So if we're thinking about using these broadly neutralizing antibodies for ADCC activity, we have to think about it differently from the people who are using these with broadly neutralizing activity. So the third IRF, and I don't have too many more slides, so we will get through this quite quickly, is to try and make those effector cells go where the virus is hiding, and in particular, potential lymphoid sanctuary sites. And Liz Connick and Pam Skinner, who lead this program, have been transducing SIV-specific cytotoxic T cells with CXCR5 to try and make those CTL go to B-cell follicle reservoirs. And so their approach is to, in a monkey model, transduce CD8 cells, expand them, check them out, and then use an ex vivo migration assay or an in vitro migration assay, and then inject into macaques and ask questions: can they actually migrate to the follicles? Can they decrease follicular virus, etc.? And so this is work that Pam Skinner has done in collaboration with Jonas Sasha and Lish Nadvolo, and they take in this migration assay rhesus PBMCs, label them with CFSE, and then drop these onto freshly prepared rhesus macaque lymph nodes, and then stain with antibodies that will help pick up anti-CD3 or CD3 or B cells, and then count and ask, where do those cells go? And the cells that are transduced with CXCR5 end up in the follicle, and if you do a ratio, a follicle to an FEF ratio, then you get more cells into the B-cell follicles as compared to extracellular. And then, excitingly, you don't even have to transduce CXCR5 at all. This Alt803 will increase the number of SIV-specific CTL in the lymph nodes. Alt803 makes these CTL, or helps these CTL get to the lymph nodes, probably by upregulation of CXCR5. And so that's sort of very exciting data. And at the moment, Liz and Pam are working with Jake Estes using RNA-scope and DNA-scope to start to look not only in rhesus macaques, but also what's happening in HIV-infected people. Finally, and we think a very exciting part of our program, is our clinical hope for HIV-infected participants. And in our grant, Kath Bollard and Gary Simon are going to do a clinical trial of autologous infused T cells with Alt803. We're building upon a platform Kath Bollard's already worked on with Dave Margolis, We're using peptides to help expand out the ones that will recognize non-escaped epitopes. And we're going to be using the best in vitro and in vivo animal models to help direct which way we're going to do this in people. And we have already started this. We've made clinical grade peptides to expand the CTL. And then we'll be looking to see whether we can get targeting of epitopes. And the clinical trial design will occur like this. We will leukapheresis patients, and then we will come in with a number of different infusions of the HIV-specific CTL, in addition with weekly Alt-803 injections, and then monitor with blood draws. We're hoping we will also put in fine needle aspirate to look at the important lymphoid reservoir. And this synergizes with all of the IRFs because we have to feed information to help Kath and Gary determine which will be the best clinical approach that we will take with people. And this is a 5-year grant. We have a number of different milestones. First couple of years, we have to sort of set everything up, establish the safety, and do the trial. And so we ambitiously are attempting to begin enrollment in November this year in Washington, D.C. And we hope that eventually we will be able to roll this type of study out, if it's successful, to other sites that participate in the Collaboratory, including potentially Brazil. So this is ongoing. So just very quickly, we have these support sections, non-human primates, reservoir characterization, regulatory affairs. The regulatory affairs basically does this incredibly important work. If you want to actually get something into people, you have to have good manufacturing processes to get those cells at the right stage, and then you have to have a whole pile of regulatory activities. Kath Bollard at the Children's National Hospital on a daily basis grows up EBV, CMV, adenovirus, and BKJC CTL for infusion into kids with cancer. Works very nicely. They ship them all over the United States, and it's all under GMP. They also are helping us with the consent, along with our community advisory board, the preparation for the IND, and the clinical process. The non-human primate support is run by James Whitney at Beth Israel with the animals at Wisconsin. And then Brad Jones runs this amazing characterization where he has leukapheresis samples that are characterized and have reservoir data from a large number of individuals. So I'd like to end there. Again, come back. There are a large number of people. There are probably 200 people working on this project altogether. We're having our annual meeting in Washington in June. And I want to really thank the funders because it requires a lot of funding. Hopefully the funding will continue. And happy to join the panel and take any questions. Thank you.